After transcription converts a DNA strand into mRNA, translation is the next step to convert the sequence into protein. Before we go into translation, let's take a closer look at the DNA strand being translated. Here we have a DNA strand that, when transcripted into mRNA, converts all of its T bases into U bases. Once this is complete, the mRNA strand goes through translation, and the following proteins are formed: methionine, tryptophan, asparagine, alanine, cysteine, and then a stop codon represented by UGA. This represents what would happen with a normal DNA strand with no mutations present, but what happens when there are mutations in the nucleotide bases? Well, there are 4 different types of point mutations that can occur in an mRNA strand that could alter the type of protein that is formed during translation. The first type of point mutation is a missense mutation. This is when one base within one codon in the mRNA strand changes to a different base and causes the resulting protein to change completely from the original intended protein. Due to the base changing from purine G to pyrimidine C, also known as transversion, the normal protein tryptophan instead translates to produce the protein serine. The second type of point mutation is a nonsense mutation. This is when a base change in the codon results in a stop codon. In other words, it causes translation to cut off earlier than it is supposed to, not allowing the remaining mRNA strand to be expressed. Here we see a mutation in the cis codon, where a transversion from pyrimidine C to purine A has led it to being coded into a stop codon, causing the mRNA to stop coding a whole codon before it is supposed to. The third type of mutation is a silent mutation. Here, a change in a base does not change or affect the protein. For example, here we see the U in our GCU codon mutated to a C, indicating a transition. However, as translation occurs, alanine is still the resulting protein, causing no disruption to our normal intended strand. The final type of point mutation is a read-through mutation. This affects the stop codon, specifically causing it to be read as a normal codon and allowing translation to occur past where it's intended to stop. When compared to our normal sequence, we see that the base U in our original stop codon has mutated to a base A. In other words, a transversion has occurred. Due to this, the original stop codon instead codes for arginine, and the ribosome continues to read our mRNA sequence.